Paper chromatography is a way to separate pigments that you can find in plants. So, why is this important? We know that plants have different types of pigments, chlorophylls, accessory pigments such as xanthophylls and carotenes. So, through the process of photosynthesis, plants use carbon dioxide and water to produce sugar and oxygen. So, how are these pigments important and how do they separate in terms of using the paper chromatography method? In this lab, we will look at the ratio factor. In fact, you will calculate the ratio factor for such pigments. And so basically, the ratio factor is the distance moved by the pigment. In this example, it moves 2.1 centimeters divided by the distance moved by solvent, in this case 2.8 centimeters. Notice that the ratio factor does not have a unit, is simply a number after you make your calculations. Now, if we were in the lab, you would actually use the spinach leaves and we would actually grind them and concentrate them onto a chromatography paper. And this is very neat because if done right, you can actually see the different colorations. Chlorophyll B, chlorophyll A separates first, then you see a yellow, bright yellow xanthophyll, and then finally a lightly orange carotene pigment. So you will calculate the ratio factors giving given these travel distances in centimeters. So for your lab report, just calculate them, place them in the boxes here in the column, and answer the following questions.